Well, hello everyone and welcome back to Science in the Age of COVID-19. Today we are super excited to welcome Dr. Alessandro Sette from La Jolla Institute for Immunology in San Diego. Dr. Sette has a long track record in studying T cells, the expression and isoforms of T cell receptor, MHC, epitope selection and presentation. His lab has developed numerous tools for use in this area, many of which I myself have used, so I am forever in your debt. Dr. Sette's lab has done a bunch of stuff with regards to SARS CoV-2. Mostly on the T cell response, which I believe is most of what he will talk about today, but also circling back to B cell response and everything in between. So Alex, take it away.
Thank you, Loren. Very nice introduction, and I'm excited to be here. Let me share my screen so that hopefully you can see the slides. Okay, oops, this is the end of the talk, so sorry about that. So as I reverse to the start of the talk, just wanted to say that the work that I'm going to present to you today has been a result of a close collaboration between the lab of Shane Crotty and my lab and Professor Daniela Weisskopf, an assistant professor here at La Jolla Institute, and Alba Grifoni, an instructor at the Institute. And then many very talented postdocs and graduate students listed here, and program managers, bioinformatics. Alex Gatto has done a fantastic job looking at the memory B cell response and so forth, and clinical collaborators at the UCSD and HLA typing from Murdoch. So this really the story starts a year ago, or a little bit over a year ago, when in February we first, it does look like a century ago, but it's almost a year ago only. This first reports of SARS CoV-2 started to come in, and at that point there was a lot of concern about whether this virus is going to be immunogenic. How hard is it going to be to induce an immune response? Is it possible to develop a vaccine? And we were interested in developing tools that would allow us to measure immune responses. There was also a lot of concern that T cells may be bad, that the acute case, severe cases may be caused by Th2 response and whatever. So in any case, this is all a theoretical discussion, because if you can't measure an immune response, you can't develop a image of immune response. And then if you develop a picture of immune response, then we can discuss whether we like a picture or not. But if there is no picture to be taken, there is no discussion to be had. So just to quickly emphasize that immune responses at the adaptive level are complex. There are antibodies made from B cells, obviously crucial to bind the virus, neutralize it, to prevent infection. And the helper T cells and the killer T cells, conversely, do not prevent an infection, but are important in terms of terminating an infection, because they can recognize infected cells, while antibodies, once the virus is inside the cells, can no longer access it. And the helper T cells are the great orchestrators, key for the development of an antibody and a killer T cell response. Also, some data in animal model that they have a direct antiviral effect. So we started interrogating the data that we had back in February And we noticed, like many others, that this new virus was very close to SARS-CoV-1, if you wish, the one had caused the previous SARS outbreak, and also to a certain extent other MERS, for example. And really the closest relative was SARS, and there was a significant amount of data that had been generated regarding characterizing immune responses to SARS-1. And so we were able to model the structure of the virus, sequence of the virus, what was known about the level of immune responses to SARS, and based on that, make predictions of what were likely targets of immune response. And we synthesized peptides that would allow us to actually test whether we could detect responses in SARS-CoV-2. And the first target, this was also, the first paper was published in March, and this Cell paper was published in May. Eventually, it was available online before that. But the take-home message that we could see robust responses in uncomplicated recent convalescence. And we studied recent convalescence for a reason. We wanted to see what does the immune response look like in a success scenario where the body has been able to control the virus without requiring hospitalization. And so we saw good helper responses, killer responses, good antibody responses. So that was good news. We actually synthesized peptides spanning the entire proteome. And you see here the proteome were about 25 different NSP and ORS and structural proteins. And we measure the CD4 and CD8 response directly ex vivo with an assay that doesn't really alter very much with reactivity because it's a short assay. So you're actually measuring the memory responses. And what we see here, that spike is a good antigen. It's a good antigen for CD4. It's a good antigen for CD8. This was good news in terms of vaccination efforts that were already ongoing, but utilized the spike antigen. So spike has it all. Has the capacity, obviously, of being targeted by neutralizing antibodies, but also target for CD4 and CD8 responses, which is not always the case. For example, in dengue, the main target of neutralizing antibody is not a particularly good target for T cells. So good news. At the same time, you see that there are other proteins, other ORs, other NSPs that are also targeted by T cell responses, which suggest that if required, these additional targets could be included and considered in vaccination efforts, and also should be certainly investigated in the sense of investigating the host pathogen interaction and immune responses. So these were the conclusions. We developed these pools of peptide that allowed to reveal robust responses in uncomplicated cases, and the good news, and S is a good antigen, but also additional targets. And then actually, this paper was quite popular. Tony Fauci showed it in Congress, waving our paper, which is going to be hard to repeat. So we stop now at this point with the convalescent, and we turn our attention to acute phase cases. And so you see here that in general, we could see good CD4 T cell responses in the convalescent, as I showed you, the black points, is whether you look at the S, M, or N, or the rest of the genome. But if you look at the acute stage, there's much more of a variation. And you see that in reality, there are some people that do not have a good CD4 T cell response in the acute phase, while others do. So we looked at this in more detail, and we naively said, okay, let's just develop a adaptive immunity index. And so we said, well, What about, for example, if you look at antibodies and you segregate people that have stronger, weaker versus stronger antibody response, and you see if there is a difference in people in these two categories, whether they are severe or mild cases. So does the antibody predict in the acute phase whether you're going to have a severe or an acute case? And it really doesn't. But what it does predict, associate with disease severity, is the CD4 T cell response. You see that people that have low CD4 T cell response, as I showed you in the previous slide, are disproportionately associated with severe disease, while people that have mild cases are disproportionately represented by people that had high CD4 T cell response. This is also true for CD8, and actually the best correlation is if you consider all three responses together. So in a way is that, yeah, and I'll elaborate this in the next slide. So in a more quantitative fashion, we looked at a number of different variables associated with CD4, CD8, and antibody responses. And what we saw, compatible with our adaptive immune response score, that basically you had in the acute phase a lot of coordination. So that CD4 and CD8 antibodies would go in concert. One thing that is known is that the age is a major risk factor in COVID. In fact, what we could see if we looked at people that are less versus less than 65 years old versus older than 65 years old, that there is a major breakdown of a communication, if you wish, in the sense that the responses in the elderly people appear to be less coordinated between CD4, CD8, and antibody responses. Another important result that came from this Motor-Baker cell paper was that the thing that correlated the most was the decrease in the naive repertoire associated with age, which is also known. So in other words, what we might be seeing here is that in older people, the naive repertoire, the repertoire of naive T cells that can respond to a new infection shrinks. And that obviously puts the immune system at a disadvantage. There is more catch up to do in expanding a adaptive response. So summarizing with SPARK, in acute phase COVID sample, we had two different studies that they came out in early and late summer, which had similar findings. And basically the bottom line is it is good to use all three branches of the adaptive immune response. Perhaps if you want to make an analogy, if comparing the SARS-CoV-2 infection to an invasion from an enemy, it's good to use your whole capacity to fight that, to use both the Army, the Navy, and the Air Force. And if you bring a coordinated response, that is most effective. And also, the other point that came out is that this response is most effective when it's happening early. So the next logical question is, okay, so we characterize this immune response in the acute phase and in early convalescence, but how long does the immune response last? Which is obviously an important point, both in terms of the legitimate concern over potential reinfection, but also in kind of extrapolating potentially to the issue of how long responses induced by vaccination will last. So we analyzed this in a study with about 200 people, 188, followed over a period of time at the end 6 to 8 months. And we measured, based on the data from the previous studies, we measured antibodies and B cells and helper T cells and killer T cells, because the previous study told us that it's really important to look at the coordinated response. So this is representative data from that paper. It came out in January. And you see here we measure responses in terms of days post-symptom onset. These are spike or RBD or neutralizing responses in a cross-sectional mode. And these are longitudinal samples where we had the same person donating 2 samples over a course of time. And you see that the responses are remarkably stable. Same for T cell responses, and I have a summary slide. Yes, that is here. So what did we see at the end of this study? First of all, I want to point out that this is the largest ever study for any acute infection, as far as we can tell. And what we see is that the antibody response is very stable, with T1/2 of several months or plateau in terms of a neutralizing titer or IgA. In the case of memory B cells, they actually tend to increase over time, over the first 3 months, and then plateau. And also relatively good T cell responses that are maintained over, excuse me, a long period of time. The other point that comes from this study is that really the immune response, whether you look at antibodies or T cells, is very variable. I mean, it varies. This is log scales, varies over several orders of magnitude. So there is this fantastic degree of heterogeneity in the responses induced by natural infection. This could be due to many different reasons. In a way, it kind of mirrors the fantastic different spread of clinical outcomes from totally asymptomatic to actual death that can follow SARS-CoV-2 infection. And as we were saying, we're very interested in investigating this further. And there are variables that may have to do with the infection itself, how much virus was the person exposed to, and so forth, for how long. Other variables that might have to do with the age, gender, sex, ethnicity, other things like that, or the genetic makeup, interferon type 1, a big one there, has been reported also the ABO, blood sugars, and other things like that. So it's a very complex picture that will have to be unraveled. At the end of the day, 8 months plus, about 90% of individuals are still positive for a number of different measures, at least 3 out of 5 of the different immune responses we measured, which is very good news. At the same time, 90% having it means 10% don't have it. And so this should be really taken seriously, in the sense that having been previously infected does not guarantee that you are now immune, and certainly is not known for how long. Also, is not known what level of immune response translates in protection. So we still, based on this study, we urge people to maintain their social distancing, masking, and so forth. And people that have previously infected also should be vaccinated to ensure and strengthen their protection. So again, an analogy that I always make is like, if I would give you a car and say, well, I probably have 90% probability that the brakes work, I think you would say, well, I don't like that. I want to have 100% probability that the brake work, and I'm going to take it into a shop to get my brake functioning for sure. Now, moving to a different aspect of the work that we have been doing, in the original first Cell paper, we noted that the negative control was not totally negative. So, of course, we included a negative control, like in any study. For us, a negative control were PBNCs that we had stored from 2015 to 2018. So there's no way that those people in California would be exposed to SARS or SARS-CoV-2. And in fact, we knew that was the case when we ran serology assays. But nevertheless, in a good fraction of the individuals, we actually saw significant T-cell reactivity. So we and others saw that in several different studies, more than 10 studies now all over the world, have clearly shown that. The hypothesis was that maybe this may be due to the fact that the negative controls with unexposed individual had been exposed to common cold coronaviruses that share homology with SARS-CoV-2. And so there may be some memory T cells that are cross-reactive. And this is important because if there is pre-existing cross-reactive memory response, that could influence the disease severity and could also influence the outcome of SARS vaccination. These are issues that are still debated to this day. To address this, we started identifying the repertoire of epitopes recognized in non-exposed donors. We identified 142 epitopes. This paper came out in Science in August. And now knowing what the epitope repertoire recognized in non-exposed individual, we could ask more direct questions. So, for example, we picked about 30 spike and 30 non-spike of these epitopes that we're seeing, the SARS-CoV-2 sequences that we were seeing being recognized in non-exposed individuals. And we made the matching homologous peptide from common cold corona. So give or take 30 epitopes from the remainder of the genome and 30 epitopes from spike, multiplied by about 4 different main common cold coronavirus. 30 times 4 is about 120. So in reality, we ended up with 129 or 124 common cold coronavirus homologs of the SARS-CoV-2 peptides that were recognized in unexposed. And then we tested them side by side, and we saw that, in fact, the common cold corona were recognized even better than this SARS-CoV-2 matching sequences, suggesting that, again, what had induced these T-cell responses in vivo was perhaps common cold coronaviruses that had induced a memory T-cell responses that then cross-reacted on SARS-CoV-2. We did do memory analysis, and I'm not going to go through the details of this slide, but essentially we showed that this response that was detected directly ex vivo was a bona fide memory response. And then we made specific T-cell lines by in vitro stimulation with the peptides, and we were able to show that there was indeed this cross-reactivity at the level of identified peptides. So the experiment here is in black, is the SARS-CoV-2 peptide that we use to generate the in vitro T-cell line. And then we interrogated those T-cells and asked whether it would recognize peptides from common cold corona viruses that were homologous and matched to that sequence. And they did. In fact, in a number of cases, they recognized the common cold corona versions even better than the SARS-CoV-2, again suggesting that the original antigen that induced that memory T cell might have been a common cold corona sequence. So this was heavily debated And because, as I was mentioning, the potential implications for vaccination and for herd immunity. And I think there was a lot of debate. Some people actually jumped on this observation to say, oh, we have pre-existing immunity in half of the people, so it's fine, we can throw away our mask and reopen the economy. And that, again, we cautioned that that was not an accurate representation because it's not known whether this pre-existing reactivity that we see has in reality a meaningful impact on responses, on disease severity or vaccination. So until that data is out, really this is still something that needs to be investigated. There are studies that are coming out that suggest that indeed, for example, a recent common cold coronavirus infection may be associated with less severe disease, which would make sense because from what we know, in order to prevent an infection, you need the antibodies. But T cells could play a role in the course of the infection if there is a degree of T cell reactivity that could modulate and make the response, the disease less severe. So in conclusion, clearly reactivity is detected in non-exposed individuals, reproducibly detected early on. At the same time, we were making this observation. Thiel's group in Germany saw the same thing. Roy de Vries in the Netherlands, Bertoletti in Singapore, in the UK. So it was really reproducibly observed. And we were able to show that this does reflect cross-reactivity, at least in some cases with common cold coronavirus. Well, how this may influence immunity and disease severity and vaccine responsiveness is still something that needs to be addressed with additional data. Now, what I showed you thus far is are the results of studies defining the epitope repertoire in non-exposed individuals. So this pre-existing immunity in an unexposed individual that can recognize SARS-CoV-2 sequences. Then we turn our attention to defining the epitope repertoire in people actually infected that had were infected with SARS-CoV-2 and had recovered from the infection. We looked at about 100 different people, 99, a span of different disease severity and ethnicities, reflective of essentially the disease burden in the San Diego area at the time we're doing the study. We developed a screening strategy where we looked for class 2, basically overlapping peptides spanning through the entire genome. For class 1, we used this approach in combination with an approach where we looked at predicted optimal peptide for 28 different class 1 alleles that represent the large majority of what is recognized in the population, patient population we were studying. So these are the results, and this was published in CellMed early this year. Here you see CD4 and CD8 response. You see the various antigens that make up the SARS-CoV-2 as different rows, and each column is a patient, a subject. So in the form of this heat map, this allows you to recognize immediately what are some of the good targets of immune responses. So you see for CD8, NSP3, S, M, N, very similar pattern for CD4. The other thing that this map does, tells you how many antigens does the average person recognize. And so the average person recognizes about 3 to 4 to 5 different antigens, or 2 to 3 to 4 to 5. In general, the response, therefore, is multi-antigenic. Then the other thing that this does is tells you which antigen you can screen to identify the specific epitope. So which antigen donor combination. So for example, if you're interested in CD8 T-cell responses against NSP4, this is a good guide to look at. This is not a good guide to look at, and so on. For NSP16, that would not be a good choice because it doesn't respond. But with this donor, it's not a good choice if you want to look at NSP3 or NSP4, but it's a great choice if you want to look at the CD8 response for NSP16 and so forth. As a result of these studies, we identified about 800 different epitopes. And what we see very conservatively, because now we know how many epitopes are recognized in the antigens that are recognized, very conservative. Each person recognizes 15 to 20 different CD4, 15 to 20 different CD8 epitopes, and also, importantly, the epitopes are different from one person to the next because different people recognize different antigens, and also because of HLA, the actual peptides recognized is going to be different from one person to the next. So this is data for the 300 or so, 280 different CD4 epitopes. Excuse me, we define lots of data. I'm not asking you to see this table, but we were able to define binding patterns and inferred HLA restriction for a good fraction of these epitopes. An important point is then how does this overlap with the epitope repertoire that we previously seen in the unexposed individual? And so we saw that by and large, we re-identified a lot of the epitopes we have previously identified in unexposed. So we see the same epitopes in unexposed and exposed people. But the vast majority of the epitopes recognized in COVID-19 convalescent people, it's new. So there is this pre-existing cross-reactive response in unexposed individuals. So once the individual is infected, yes, those are going to be activated as well, but it will generate its own repertoire, a new repertoire of additional T cells reactivity, much broader than what you would see in unexposed individuals. We looked at various different correlations. Not surprisingly, as a result of what I was telling you a minute ago, there is no correlation between what is recognized and the degree of common cold corona homology. The one thing that does correlate is HLA binding. Switching for a second to CD8 responses, we see here a phenomenon that we've seen before also in, for example, in the case of dengue. So when you look at different epitopes recognized by different HLA alleles, you see different patterns. So for example, A1 has very few epitopes, relatively speaking, but when there is an epitope, you see here the magnitude, it's a very good response. And the reverse, for example, is seen in A26 or A6801. And it's a lot of epitopes, but the response per epitope is not that good. This is a log scale. And so, and then you have some that are not very good by either criteria, either frequency or magnitude of response. So there is a variety of different HLA reactivity. And this will remain to be seen whether this correlates with disease susceptibility or not. Obviously, this data set doesn't have a power to address that. Based on this identification of different epitopes, we refined our peptide pools that are being looked at and have been produced. And so we now have this experimentally defined epitope pools that have p-values of 10 to the minus 9 in discriminating exposed and non-exposed individuals, for example. And we have been sending this and the previous mega pools very openly to a number of different labs all over the world. So in conclusions, we identify 280 CD4, 520 CD8 epitopes. The CD4 epitopes are promiscuous, they bind, the dominant ones bind multiple alleles. CD8 epitopes, there is this phenomenon of different HLA alleles having different repertoire size, breadth of epitope repertoire, which I'm going to get back to it. There are clear immunodominant regions. I didn't show you the data for CD4 responses, and RBD is relatively epitope poor, but regions to the left and to the right are rich in CD4 epitopes. Common cold corona conservation doesn't really correlate with the epitope repertoire seen in the course of SARS-CoV-2 infection. HLA binding is a major correlate, and pools of epitopes that have been designed on the basis of these results really facilitate measures of T-cell responses. Now, let me close with a few slides regarding the variants of concern. So we looked at B117, B1351, P1, and California variants. All are associated with increased transmissibility, and in particular, mutations in the S antigen have been of particular concern, and it has been shown that these are associated with a loss or a reduction in the capacity of being neutralized or bound by monoclonal or monoclonal antibodies. The data that is coming out regarding vaccine efficacy is that in some cases, vaccine efficacy is retained. So B117 is not really that strongly impacted at the serological antibody response level, and vaccine works against these variants quite well. There is more concern for other variants, and there is a correlation. These variants are associated with a greater decrease in capacity of being neutralized, and the vaccines have been reported to have a decreased efficacy. Importantly, efficacy is retained thus far, what we've seen in general, in terms of protection from death or severe disease. So we can interpret this data or speculate in the context of what we know about T-cell responses. There is data from the paper I was referring to, similar data from Bertoletti's group in Singapore, where we see that early CD4 and CD8 T-cell responses are associated with milder disease. There's other data that I did not show you, but that shows that persons with agamma-globulinemia, so a genetic defect where they don't have antibodies, or patients that have undergone B-cell depletion because of other pathology, nevertheless experience uncomplicated COVID infections. And of course, that CD4 and CD8 response is induced, as I show you, in after infection or vaccination. So we thought it was important to understand whether the various variants had also, besides the decrease in antibody response, a decrease in T-cell reactivity. So we generated peptide pools spanning the entire genome of the different variants, and we tested them for reactivity with PBNCs for either convalescent people or people that had received either Moderna or Pfizer vaccination after the second dose. And this is important. So this is done, obviously, the vaccine are based on the ancestral Wuhan sequence, and also these convalescents were from a time where the variants were not infected with ancestral Wuhan or similar sequence. So what does it look like? You see here the response against the ancestral peptide pools, or the peptide pools representing the various different variants. And what we see is that essentially there is very similar reactivity. Whether you look at the activation-induced marker assay for CD4 or CD8, we also used a different assay, which is the ELISpot assay, similar results by and large, and the response was also all as expected, interferon gamma mediated, nothing in terms of Th2 for IL-5. We so the pattern of T-cell reactivity was maintained. We also looked at the titration of the different pools to address whether perhaps, despite the fact that there was very similar reactivity, there may be a decrease in the avidity of a T cell, and very, very similar avidities were detected here. So this is for vaccinees, for spike variants in the convalescents. Of course, convalescent people don't just see spike, so we looked at all other antigens, and this is the sum reactivity against all antigens. Again, it's very similar reactivity. What about vaccinees here? The Moderna are triangles, and Spike are, and Pfizer are circles. Same picture, whether you look at CD4 or CD8 by the AIM assay, there is a very similar reactivity. In the case of the ELISpot assay, once again, mostly gamma, a little bit of IL-5, but if you actually calculate on a per donor basis, the fraction of a response that is mediated by Th1 representative interferon gamma cytokine, it's in the 80 to 90%. And again, similar values in terms of estimating the avidity of responses by performing a titration of the spike pools. So I'm showing you this slide again to emphasize that this makes sense, because the response for the both CD8 and CD4 response is very multi-antigenic, and there are many different epitopes that are recognized in each individual. In fact, when we perform an analysis and we looked at all these 800 peptides that are experimentally defined as being recognized in humans in the context of infection, we asked the question, how many of those were totally conserved or had a single mutation or multiple mutation or deleted residues or eliminated altogether because of a stop codon? And you can express the data for CD4 or CD8, whether you look at all proteins or spike, and you can look at either the percent in terms of number of epitopes or the percent of a total response associated with those epitopes. And without going through each combination, basically you can see that about 90% of a CD4 T cell response is conserved in the variants, and 95 to 98% of a CD8 T cell response is conserved. So that, and well, I'll skip over this slide in the interest of time. So to summarize this last section of the talk, we evaluated the common variants of concerns for CD4 and CD8 responses in convalescent and vaccinees. The T cell responses were largely unaffected. The bioinformatic analysis on known epitopes confirmed that that finding. Of course, I want to emphasize that this is at the population level. It could perfectly be true that some individuals may happen just to recognize some epitopes that are impacted and where they might be outliers in the response in that respect. But at the population level, really remarkably not affected. So bringing it back with what I was mentioning earlier in terms of vaccination, from what we see is that it's likely that the antibody response against the variants may be affected to variable degrees depending on the variant and a vaccine, and it's associated with a reduced capacity to prevent infection. But if, as it seems, T-cell responses are largely unaffected by the variants, it's presumable, it's plausible that those T-cell responses would be able to modulate disease severity. And this fits with what we see in terms of the vaccination still protecting from severe disease and death. This also suggested second generation vaccine that would incorporate additional antigen or epitopes can be considered as of interest to broaden and enhance the T-cell responses. So in conclusion, we've seen robust CD4 and CD8 T-cell responses in convalescent uncomplicated cases. Reactivity is also detected in non-exposed subjects, and this was reconciled in at least some cases with cross-reactivity with common cold coronavirus. We also defined the epitope repertoire in COVID patients for both CD4 and CD8 responses. The studies in acute and severe cases highlight that speed and coordination of immune responses is key. We've shown that T-cell responses induced by natural infection are durable over at least 6 months. Obviously, we and several others are looking at how durable the responses induced by vaccination are going to be. It's early because there's not a lot of various vaccines that are approved for emergency use have not been approved for very long, so we'll have to wait to have a more abundant data set regarding that respect. And finally, there's a relatively negligible impact of variants of T-cell response. And with that, I will stop share and thank you for your attention. And I'll be glad to answer a few questions.
Alessandro, that was a fantastic talk. I can tell you, I learned a lot about adaptive immunity in the past 1 hour, and I'm sure the audience did as well. So audience, just another reminder, please keep your questions coming. You can ask your own question, or we would be more than happy to read out your questions for you. So as the audience is getting ready, let me ask you a question. So you very elegantly showed that T cells from the sera of convalescent individuals and also vaccine recipients are able to neutralize variants as well. But have you seen the reverse? I mean, are T cells from individuals affected by the variants, are they able to cross-neutralize the ancestral strain?
So that's a very good question. We don't know. We plan to do those experiments as we will be able to secure PBNC from people that we know have been infected with particular variants, so we can do that experiment in reverse if you wish. Very encouragingly, there is data from Segal's group in South Africa that at antibody level shows that that is the case. And in fact, so while the antibodies induced by the more ancestral sequences lose activity against the, for example, a variant originally described in South Africa, a person infected with that variant, the sera will cross-neutralize not only this South African variant, but also the other variants, which is just great. It's great data to hear. Should I answer some of the questions in the chat?
if you want to, we can also read them.
You tell me.
Sure. Why don't you? Why don't you just start going through and reading them out?
Right. So Dieter asked any correlation between S peptides and glycosylation. We looked early on. We did not see a lot of correlation. We need to look more. And in general, T cells do not like to recognize glycosylated peptides, but when the glycosylation is removed, that could change basically an N to a D. So it's worth looking into that a little bit more. There was another question. I'm curious whether we can predict or infer from this data about duration of protection from the mRNA vaccines. Will we need booster after 6 to 8 months? So let me take out my crystal ball. No, but it's a good question. And so let me answer by pointing out a few things that are potentially relevant. Of course, we don't know how durable responses induced by the vaccines are going to be, and eventually we will need to wait 6 to 8 months to know if they last 6 to 8 months. But a couple of things is that in general, the response induced by vaccination is higher than the response induced by natural infection. The other point, which is really very encouraging in my mind, is remember there was this several logs variability in the immune response induced by natural infection. The response induced by vaccination is a lot tighter. There's a lot less variation. Now, based on that, I am optimistic. You're starting from a higher level and you're starting from a tighter, less variation. I'm optimistic that this would last and potentially outlast the duration of immunity induced by natural infection. But the reality is we don't know. So we'll have to wait. But right now there is no reason to suspect that you would need a booster every 6 to 8 months. Obviously the variants may complicate that equation, but again, there are several companies working already on generating boosters with the variant spikes, which or generating broader T cell responses, which would be encouraging.
So, Alex, I was really happy to see your data on the variants because over the past couple of months, I've checked into T cell responses, and and there's scattered reports. Like I just looked at one briefly, didn't read it, but that the N501Y mutation from B117 does hamper display on MHC class 2. But I mean, as you say, maybe we're just protected by the redundancy of our response, and so if we lose a bit of that, it's no big deal. So I mean that was very comforting. So
right, yeah. So and let me elaborate on that. Yeah. So there are a couple of reports that show that there are instances in which the mutation found in some variant decreases activity of some epitope. And that's fine. I mean, that is not a problem. Obviously, if you look at enough mutation and enough HLAs, there are about a few thousand different HLAs, you will find. And in some cases, has been shown quite clearly that for that particular HLA, that particular epitope is decreased. But again, we took an approach of looking at the population level, and because of the redundancies, that particular epitope undoubtedly could be decreased, but at the end, at the population level, that doesn't seem to play a role, at least with the variants we've seen. I would also point out that there is really no data in any system that I know of that an acute virus has mutation that escape HLA recognition. The data that is very strong is, for example, in HIV, where it's very clear that there are mutations, but there's a key difference. It's a chronic virus. So obviously, if the virus is in the body for years, then certainly there are mutations that accumulate that can escape. The other point that is important point to consider, again, and we're now just speculating or chatting about general principles, but it's important to consider that the breadth of responses there is always a cost for the virus. So you could envision, like, okay, we're going to have the virus, the crazy virus that has 800 mutations every single epitope, but mutations have a cost. So the virus has evolved to optimally reproduce, and so you start to introduce mutations that has a cost on the virus. In fact, what you see often is if there is a mutation that is there because of escaping immune pressure, if you remove that immune pressure, the virus goes back to the way it was. And so that it would be difficult to envision for me that the virus would retain all the mutation against all different HLAs if it's not replicating in a person that has that HLA.
Yeah. I mean, there do seem to be very rare mutations that do both, that both improve fitness, like ACE2 binding, and assist in immune escape. But I agree.
No, and that's certainly that's for spike is certainly true in antibody responses, because also the region that is important for binding and infecting is also this region that is important as a target from neutralizing antibodies. So that's obviously that's a different unfortunate situation.
Yeah. Hey, I had a really technical question. So you're talking about second generation vaccines potentially introducing different epitopes. Do you see those as peptides, the short peptides that are added to the cocktail, or do you see that as changing the sequence of the protein such that when it's carved up, you get good T cell epitopes while also eliciting a robust antibody response?
I think that all approaches are worth considering. So clearly, to make a vaccine based on a variant that induces a more neutralizing antibody response brought into as variants is a great approach. In parallel, people are considering adding additional antigens that are more conserved. So N, for example, or M, or NSP3, or whatever, but it could be delivered alongside the spike, perfectly fine. Or another option is to take chunks, to use a technical term, of proteins that are good targets for T cells and just put them together in some kind of cocktail and whatever. I think all three are being considered and all three are legitimate.
Yeah, I can't remember which ones, but I know at least one, maybe two of the first generation vaccines said that they were including short peptides to be MHC epitopes, but I lost track of which ones it was and if they're even approved.
No, I don't think there is anything that is authorized that has short peptides, but
Do I have time for one last technical question or do?
One last technical question.
And I'll keep it short, I promise. So thanks to the work of Professor Akiko Iwazaki and others, we've learned that COVID-19 manifests itself differently in different populations. For example, male evoke a much different immune response than females. And there's also an age difference. So in your cohort, what was the distribution of, the sex distribution and age distribution?
Yeah, so the sex distribution is fairly balanced. And in fact, in the Dan paper in Science about duration of immunity, there is some data showing differences in gender. There is a significant difference, which is by no means explain the tremendous difference over several logs. We are interested in looking at this. The problem is that you need a fairly sizable cohort. So we're trying to assemble also with some degree of targeted recruitment, a balanced cohort, ideally spanning through different ages and different ethnicities, Blacks, Asian, Caucasians, Hispanics, of course, sex, and different disease severity from asymptomatic to hospitalized. So problem is that certain categories are much more difficult to come up with. The hospitalized ICU cases are, thanks God, much harder to find than mild asymptomatic infections. And so we're assembling this data, but we don't have a comprehensive enough yet cohort to be able to answer and dissect the different variables, right? Because if someone is, if you see a particular type of immune response in a Black male subject of 65 years old with intermediate disease, what does that mean unless you have you can compare it to similar cohorts in the other variables. So that's something we're certainly very interested in doing.
Thank you.
Yeah, Alex, thank you so much for your time and amazing research.
My pleasure.
Stay safe and keep up the good work.
Thank you. Bye, everybody.
Yep.
Thank you.
Okay, and Matt, can you bring up the slide for next week? Yep, next week we'll have Professor Laura Vella from Children's Hospital of Philadelphia. So join us next week, same time. All right, bye-bye, everyone.